Good afternoon. I'm Bruce Mann, the Research and Education Lead for the Victorian Comprehensive Cancer Centre and MC for today's Grand Rounds. This is a fortnightly event showcasing research and developments in breast cancer related fields. Before we start, I'd like to acknowledge the traditional custodians of the land on which we're all located and pay respects to elders past, present, and extend respect to other Indigenous Australians present online. We've had a number of these. I think most people are aware of how it works. Questions, shortly I'll introduce our speaker, Caleb. Questions can either be using chat or using the Q&A function, and we will be able to have actual real questions and answers at the end. So it is my pleasure to introduce our speaker. Dr. Caleb Dawson recently completed his PhD with Professor Jane Visvader, Dr. Anne Rios and Professor Jeff Lindemann at the Hall Institute. He studied mammary stem cells and resident immune cells in the breast. And what he will be presenting today is some of the work on mammary intra-epithelial immune cells revealed by 3D confocal imaging. I have heard about this work, and I think we've seen some of the early work two or three years ago. I'm very pleased that Caleb's agreed to present, and I'll hand over to him to show us what he discovered in his PhD. Thank you, Caleb.
Thank you, Bruce. And thank you for the opportunity to talk to you today and present my work. I'll just figure out how to share everything and make sure. Okay. And get a laser pointer so I can point things out for you. All right. So thanks everyone for joining in today. I'm a postdoc in Jane and Jeff's breast cancer lab at the Walter and Eliza Hall Institute. And I finished my PhD earlier this year. And I was using 3D confocal imaging to look at the mammary gland in the mouse mostly, and the development of that and how the gland interacts with the immune system with a focus on resident macrophages. And the work that I'll show you today was published earlier this year, which was really exciting for us, and I'm really happy to be able to share that with you today. I'll give you an overview of mammary gland development, and then I'll introduce tissue-resident macrophages and their diversity of phenotypes and functions in various organs, and then tell you about the new population of ductal macrophages that we found and characterization of their features and functions. The breast tissue is a really incredible organ that primarily develops after birth. So in embryonic development, we get formation of a rudimentary ductal tree, and then this doesn't grow too much until the onset of puberty. The timing here is in mice, which I'll be talking about mostly today. With the induction of proliferation at the ends of the ducts in terminal end buds, which drive the growth and branching of the gland into adulthood to form an elaborate ductal tree. And then in pregnancy, this tree blooms to form spherical alveoli that produce milk. And then when the milk is no longer needed, the milk-producing cells die during weaning in a process of involution, which remodels the gland back to the resting ductal state. And we can do 3D imaging of all of these stages to look at the individual cells in there, how they're arranged and shaped, and how they interact with various cell types like the immune system. At a smaller scale, the mammary ducts are made of an epithelial bilayer with an outer layer of elongated myoepithelial cells that are contractile, and an inner layer of luminal cells that mature in lactation to produce milk. And so we use this 3D confocal imaging technique, which was set up by Anne Rios, an ex-postdoc in the lab. And this involves fixation of whole tissue and then whole mount immunostaining, followed by tissue clearing and then mounting for 3D confocal imaging. And you can see here that we can image large areas of mammary duct, standing for the different cell types like the inner cells here and the outer cells, and then look at the stroma that surrounds that with the blood vessels in this case. And this talk will also focus on the resident macrophages of the mammary gland. And macrophages develop from circulating monocytes that infiltrate into tissues to generate macrophages that are identified by these surface markers by flow cytometry. Traditionally, people have compartmentalized macrophages into pro-inflammatory or M1 phenotype and an anti-inflammatory or wound healing M2 type. But this is a little bit restricted to the in vitro or pathogenic situations. Whereas under homeostatic conditions, there are a multitude of homeostatic factors that are unique to each tissue and niche within tissue that program macrophages into a vast array of phenotypes and functions. With some have the ability to self-renew, others are multinucleate, and really interesting features like that. There are some universal functions of macrophages like tissue maintenance and repair, phagocytosis, both of pathogens and of apoptotic cells, and they can regulate immune responses as well. So here I'm just showing tissue-resident macrophages with some generalized functions, but these are present within all tissues of the body. And within each of these tissues, there are unique populations and sometimes multiple populations per tissue niche. And these all have very specialized functions depending on their location, such as synapse pruning by microglia in the brain, or electrical conduction by macrophages in the heart. They also have diversity in their origin. So there are three waves of macrophage development in mouse, early mouse life. So in the embryonic development, we have a wave of yolk sac macrophage progenitors that disseminate throughout the body and give rise to all of the early macrophages. Many of these are replaced by the later waves, but in organs like the brain, where the blood-brain barrier is set up early on, these macrophages remain yolk sac derived for the life of the animal. Then later on in embryonic development, we have the fetal liver wave, which seeds most tissue-resident macrophages in all organs for the rest of the life, like in the lung, the liver, and the epidermis. And these survive throughout the life of the animal. And then we have a later wave of the bone marrow-derived macrophages after birth. And these contribute to high turnover populations of macrophages like in the gut or the heart. And so in these organs, the embryonic-derived macrophages die over time, and these are replaced by bone marrow macrophages. But all of these, it's not clear what the functional differences of all of these are. Yeah, so much more work is needed to be done in deciphering what the functional consequences of their origin is. And then this all relates to cancer, because under homeostasis, we have a little contribution of the circulating monocytes in the adult to these resident populations, which largely self-renew. And there are diverse populations with homeostatic functions like repair, maintenance, surveillance, and immune regulation. But then in cancer, there is a heightened infiltration of monocytes to give rise to particular subtypes. And so the balance of different types of macrophages is changed. And these are skewed then towards programs like wound healing, metastasis, angiogenesis, and immune suppression. So we're not great in the context of cancer. Now, in the mammary gland, the general functions of macrophages have been studied quite extensively. They're important for regulation of alveogenesis in pregnancy. They're essential for the remodeling phase during involution. They have been seen to be closely interacting with the epithelium, and they promote growth and branching in morphogenesis in puberty. and they're situated within the terminal end buds where there are apoptotic cells that they clear away. And they're also known to promote stem cell activity. And so this is an early study showing that macrophages promote mammary growth during puberty. So this is the normal control with the gland growing through the fat pad here. But in mice without CSF1, which is an essential growth factor for macrophage differentiation, the mammary gland growth is stunted and doesn't progress through the fat pad at this stage. On the other hand, T cells negatively regulate mammary gland growth. So this is the wild-type control here, with the gland progressing through the fat pad. But then when T cells are lost, this growth is accelerated. And then in this model, this is affecting dendritic cells with the control here, the gland growing normally. And when dendritic cells in this model activate T cell immune responses, this suppresses growth of the mammary gland. So there's a really complex interplay of the immune system to promote and regulate growth of the mammary gland. But it's not really known whether there are multiple populations of macrophages with specialized functions and what the mechanisms are by which macrophages carry out these functions. Recently, there was one study to come out recognizing that there's a population of macrophages labeled with F480 here that are positive for CD206, the surface marker. And in mice with reduced numbers of embryonic macrophages, these cells are missing. So it's thought that these macrophages are a special embryonic derived macrophage. And in the wild type mice, you can see the mouse mammary gland progressing through the fat pad here. But in these mice without these macrophages, the growth is stunted. So it looks like, in terms of promotion of mammary gland growth and branching in puberty, that the embryonic macrophages might be responsible here. But not too much more is known than this. And the reason why I'm showing you all of this is that early on in my PhD, I was doing imaging of immune cell interaction with the mammary gland. So here, ducts labeled with F-actin and immune cells with CD45 in yellow. And looking at a thin optical section through this image, you can appreciate that there's some CD45 staining within the duct. And then looking at a slightly thicker optical section, these cells start to take shape as these dendritic shaped cells within the epithelium. So we were really fascinated as to what these cells might be and whether they might have a role in regulating mammary gland growth and remodeling. So I did more imaging of these cells and found that they express highly this marker MHC2, which is an antigen presentation molecule expressed on macrophages and dendritic cells. And this showed that these cells, this here is an image through the duct wall of the gland, of a virgin gland, showing the inner luminal layer here and the outer myoepithelial layer here. And these cells are sandwiched directly between the two layers of the duct wall. And this really distinct location allowed us to look at fluorescent reporter expression in this location. And so here we looked at CSF1R, which is expressed by macrophages, the receptor for the macrophage growth factor, and they express this, so they're macrophages. They also express CD11C, which is expressed on all dendritic cells and some macrophage populations. On the other hand, we labeled them with CD11B, which stained the cells outside the duct, but not the macrophages inside the duct. And CD11B is expressed on most macrophage populations, not all, and then also some dendritic cell populations. So this told us that these ductal cells are macrophages, and so we call them ductal macrophages or DMs. And we can get a clearer view of these cells by using 3D rendering. So here is a tissue stained with keratin 5 to label the myoepithelial cells, the outer layer, and MHC2 to label the DMs. And what this is we've digitally cut off the front half of the duct so that we're just looking at the inner surface. And this reveals the network of the DMs, they're regularly spaced across the epithelium, across the whole gland. And this is also the case in lactation, where the DMs interact really closely with the myoepithelial cells that wrap around the alveoli. And they also appear to be present in human tissue. There have been some sightings of epithelial-associated immune cells in the past, but it hasn't been determined whether intra-epithelial cells were macrophages. It was not clear early on, and whether they are a distinct population with unique functions. And so we really wanted to figure out what these cells were and what they're doing. And to tackle this, we tried to isolate them by flow cytometry using the information we found by imaging. So we isolate macrophages using markers CD64 and F480, and then tried to separate these out using CD11B and CD11C. And we did find a really nice population of CD11C positive macrophages that looked like these DMs. And they are MHC2 high, which also fits with the imaging. And so we thought that maybe these are the stromal macrophages, whereas these are the ductal macrophages. And now to confirm this, I did flow cytometry on whole mouse mammary glands, where we see this proportion of DMs, and then in fat pads cleared of the epithelium, so that there are no ducts left. And here we get a complete absence of DMs. On the other hand, when we enrich for the mammary ducts, we get an enrichment for DMs. So now we know that we have a pure population of DMs by flow cytometry, and that these are exclusively located within the epithelium. We also use this FACT strategy to look at other reporter mice and found that CX3CR1 is really highly expressed by DMs and not highly by the stromal macrophages. And so we use this for 3D imaging. Now in this image, we stained with keratin 5 to label the myoepithelial cells. And looking at the outside of the duct, the basal layer, which is overlying the macrophages, obscures the DM network. Whereas when we look at the inner surface, the DM network is revealed here, these regularly spaced DMs. And then looking at the full volume of the duct with just the DMs shows just how frequent these cells are. I quantified their distribution across the whole gland from the nipple area through to the distal area and also in lactation. And there are some minor differences in their frequency, but overall, they're quite regularly spaced with a similar frequency throughout all of development. And now we had determined their phenotype and their morphology by 3D imaging, but we wanted to know how they dynamically interact with the epithelium. And to do that, we turned to intravital imaging. But intravital imaging of the normal mouse mammary gland is really challenging because there's so much fat that overlies the gland that it's hard to image through. There are a few instances of this. This paper that looked at early hyperplasia to look at cell migration out of the ducts. And this paper that looked at growth in puberty, looking at the migration of cells. But the very few studies that use this just show how challenging it is to get this technique up and running for the normal mammary gland, even though it's been used extensively for breast cancer. So we worked with Scott Mueller from the Doherty Institute to develop a new technique for in vivo imaging of the mammary gland. And this had some really good strengths, like we could image for up to 12 hours. We have access to an entire mammary gland, it's shown here. And the gland is enclosed in a sealed chamber, which helps to image in a physiological environment. And we can get really high resolution imaging, shown here with the epithelial cells and labeling of immune cells with CD45, and then collagen surrounding the duct. I use this then to look at the DMs and see how they behave. This is a mouse that expresses GFP in the mammary epithelial cells of the inner layer, and then labeling with an MHC2 antibody. And this showed that the DMs don't migrate around the duct, but they move dendrites back and forth to continuously monitor. In contrast to the DMs, the stromal cells migrate quite rapidly around the duct. And we can quantify this by cell tracking, showing that the stromal cells migrate more extensively, whereas the DMs are stationary, but their dendrites move back and forth. And then looking at how they cover the epithelium using cumulative signal, we can add the signal over time and then look at how much they cover the duct. And then only within a few hours, they cover almost the entire duct, and they're regular spacing over the whole duct regions from the nipple through to the distal regions. Tells us that within just a few hours, they will probably contact every single cell within the mammary gland. So now we want to get more into the specific characteristics of these cells. Are they long-lived? Are they embryonic or adult-derived? And do they share characteristics with other resident macrophages in other organs? So we tackled the longevity question using bone marrow chimeras. And this involves radiation of the mouse while shielding the mammary glands, the fourth and fifth glands, with a lead shield to prevent damage. And then transplanting of fluorescent bone marrow, and then waiting for engraftment. And then what this allows us to do is to look at how this fluorescent bone marrow contributes to the immune cells within these mammary glands. Now this is normalized to monocytes, which are continuously replaced, and will show constant replenishment from these fluorescent bone marrow. Neutrophils are also constantly replenished, so show equal replenishment by the fluorescent bone marrow, whereas the mammary macrophage populations are more long-lived. And the DMs here, only 30% were replaced, which results in a half-life of around 5 months. So these DMs are indeed long-lived resident macrophages. And now to address their origin, we used a model published last year, the MS4A3 mouse. And this takes advantage of the fact that in the adult, there is this progenitor of granulocytes and monocytes from which all adult macrophages are derived. And so lineage tracing using this gene is able to label all adult macrophages. But the embryonic macrophage precursors take a primitive route that bypasses this, going straight to monocytes, and so they're unlabeled. And so we can use MS4A3 pre to activate a fluorescent reporter only in the bone marrow macrophages, but not the embryonic cells. And so we worked with this team who previously used this model to determine the origin of all of these various macrophage populations. And so in populations like in the dermis or the intestine, they are initially not labeled because they're embryonic, but they're rapidly replaced by these fluorescent cells from the bone marrow. But other populations like microglia in the brain are embryonic for life, and so they're unlabeled. So working with this team, we showed that at 4 weeks before puberty, most of the DMs are embryonic, being unlabeled. But then as the gland grows during puberty, we get a really striking increase of fluorescently labeled DMs, showing that during puberty, they come from the bone marrow and infiltrate into the gland. And then this stabilizes in adulthood. The stromal macrophages show a more constant replacement, irrespective of the developmental stage of the mammary gland. And this infiltration of DMs during puberty is in parallel to a large increase in their frequency. So we wanted to know now, going back to imaging, where are these embryonic precursors and how do they spread throughout the gland? So I did 3D imaging in late embryonic development, and this is imaging of an entire mammary gland at this early stage. And then looking at an optical section from this with the macrophage reporter CX3CR1, revealed that there's around a single macrophage in each tip of the gland at this stage. Whereas in the nipple region, there were no intra-epithelial macrophages. Now looking 4 days after birth, imaging the entire gland here and then looking at the different regions of the gland, the macrophages are more widespread within the epithelium, at the distal region. In the mid region, there are a few, but then even less at the nipple region. And then looking a few days later, 1 week after birth, in the distal region, they're more widespread again, and they're starting to take on the dendritic appearance of the adult macrophages. And we also see them widespread in the nipple and in the mid region. And then they start to upregulate MHC2 here, similar to the adult macrophages. So one hypothesis for this is that the embryonic precursors enter into the tips of the gland during embryonic development and then slowly proliferate and migrate down towards the nipple to establish the population. But then as the gland grows rapidly through puberty, the bone marrow monocytes come in and give rise to most of the DMs in adulthood. So one way this might work is that the DMs can only enter in when the epithelial niche is accessible. So in embryonic development, the niche is accessible by monocytes which can come in and seed these precursor macrophages. But then after birth, this is sealed off and the monocytes can't enter in to give rise to macrophages. So they just slowly migrate and proliferate throughout the gland. But then in puberty, we get the formation of terminal end buds, which have a discontinuous basement membrane. And this may allow infiltration of the monocytes to give rise to these bone marrow derived DMs. But then we know that in adulthood, they have a slow turnover. So in adulthood, we get a setting up of this barrier again. So the infiltration of the monocytes is slow. So far, I've shown you that DMs are a unique population of macrophages expressing CD11C and CXOCR1, but a CD11B negative, and that they're long-lived and monitor the epithelium. And then using the mouse models of lineage tracing, showing that they're embryonic before puberty, but then bone marrow derived as the gland grows. But we really need to get into how they're regulated and what their functions might be. And to gain clues into this, we've used RNA sequencing. So I sorted the DMs, the stromal macrophages and dendritic cell populations, and then showing the differences here using an MDS plot. And this work was done by Bupinder, Gordon, and Luke, and showed that the DMs are really unique. They're different from the other macrophages and the dendritic cell populations. And looking at pathway enrichment showed that they are really highly enriched for lysosome genes, which could be involved with a phagocytosis function. When we compared DMs to stromal macrophages, this revealed an enrichment for Notch signaling. And Notch contributes to cell differentiation and functions through direct contact, direct cell-cell contact. So to check whether Notch signaling was actually active in the DMs, we used a mouse model which expresses GFP in cells with active Notch signaling. So this is a destabilized GFP downstream of a Notch target gene. So this GFP is only expressed during active Notch signaling, but then is degraded when Notch signaling finishes. And the DMs here were the only cell, the only myeloid cell in the gland that had this active Notch signature. Now to see what the significance of this was, we looked at DMs where we had blocked Notch signaling. And we did this by knocking out RBPJ, which is a Notch-affected transcription factor. And we knocked it out using CD11C Cree, so it is just blocked in the DMs and dendritic cells. In the control mice, we have the DMC here, which are CD11C positive, and these nicely have downregulated CD11B. But in the cells where we block Notch signaling, we have DMs here. They have slightly lower CD11C expression, and they fail to downregulate CD11B. So it looks like Notch enables their maturation. And now using 3D imaging to look at their location, the regular spacing in the control glands is quite similar to that in the notch blocked glands. So it seems like notch doesn't affect their localization, but once they enter into the epithelium, they receive notch signaling that allows them to mature and downregulate CD11B. And now to see what their function might be in mammary gland development, I used flow cytometry to track their frequency throughout all of these stages, from virgin mice through pregnancy into lactation and involution. And what I saw was this really striking increase in DM frequency through pregnancy into lactation, where they really are the dominant immune cell in this stage. And this proliferation, this was due to proliferation, because when we labeled with EDU, which is incorporated into dividing cells, this really well labeled the DMs in pregnancy, but not in virgin mice. But what I showed earlier was that the density of the DMs on the epithelium is actually similar throughout this whole development. So what we think is that this proliferation enables the DMs to grow in parallel with the expansion of the epithelium to maintain their density. So we wanted to see what their function might be, especially during involution, because at this stage, all of the milk-producing cells die and have to be cleared away. And we thought that because there are so many DMs at this stage, they could have a really key function. So 3D imaging at lactation showed that the DMs have this intra-epithelial localization. And here the gland is stained with F-actin, which is labeling the milk-producing cells here in the center of the alveolus. But then at 4 days involution, this is when the alveoli are collapsing and the milk-producing cells dying. The dying cell labeled here with cleaved caspase 3. We saw infiltration of the DMs into the lumen. We confirmed that these infiltrating cells were DMs because they express CX3CL1. And so we thought that maybe these DMs are sitting on the outside of the alveoli and are ready to phagocytize all of the dying alveolar cells really rapidly to facilitate remodeling. And to get a clearer picture of this, we used the L5 reporter mice, which label the milk-producing cells, and then looked at 3 days involution. And this is before the alveoli collapse, so it's easier to get a nicer picture of what's going on. And so we zoom in on this area here. This is the 3D zoom, and then optical sections of this region. And these alveolar cells here have reduced GFP, presumably because they are dying. And what we saw was that the DM MHC2 signal is enveloping these cells. And we also saw presence of 2 nuclei within these phagocytized cells, which is a really distinct feature of the alveolar cells, which are binucleated. And this was described by the lab a few years ago. And so now we wanted to know what the functional significance of this phagocytosis is. Is it really essential for the remodeling of the gland? So to tackle this, I used the CD11C DTR model. And treatment of these mice with diphtheria toxin causes the toxin to bind to cells expressing this receptor. And so in this case, treatment with the toxin binds to all CD11C positive cells and causes them to be depleted. And I treated these mice at weaning to deplete dendritic cells and DMs at this stage. And then collected at 4 days of involution to assess how the remodeling has been effective. In the control mice, we get nice infiltration of the DMs and phagocytosis of the apoptotic cells. And then in diphtheria toxin treated mice, get really nice depletion of the DMs. And you can see the apoptotic cells here that haven't been cleared away. And this really drastically affected the remodeling of the gland. In the control, we see the lumen collapsing as it returns back to a ductal state. Whereas in the depleted glands, the lumen remain open and filled with milk and dying cells. But this was a little bit limited because we're also depleting dendritic cells. And so to tease apart whether dendritic cells might be responsible for this, we took an approach to deplete only macrophages. The problem with this is that it will target both the stromal and the ductal macrophages. So we took advantage of the fact that around weaning, there are many, many more DMs than stromal macrophages. So we treated with an anti-CSF1R antibody, which depletes the macrophage growth factor, resulting in death of all macrophages. And treating at this time had a preferential effect on DMs, which is what we wanted. So the SNs here have some effect, but the DMs here are really completely depleted. And this is the control gland here showing nice collapse of the alveolar lumen. And then here in the treated mice, you get prolonged alveolar opening here without collapse. So these two approaches with the DTR and the antibody treatment both target the DMs, not in a completely specific way, but we think that with both of these approaches, we've really strongly shown that the DMs are really critical for this remodeling phase during evolution. And so going back to our overview, our discovery of DMs has pinpointed a population that is really critical for the involution process, and also explains one of the populations that is so closely associated with the epithelium. Other aspects that we think might be really promising to look at with these cells is the regulation of elongation, which is affected in mice without macrophages, but it's not known which populations are responsible. And also promotion of stem cell activity because of the direct contact between macrophages and the myoepithelial cells. We think this is a really enticing avenue to look at. We also wanted to look at cancer because there are some old studies that are looking at macrophages throughout tumour development. So, and this shows a real similarity to the development of the ductal macrophages. So here we've got staining with CD11B with CD11B positive and low macrophage populations. And throughout tumourigenesis in this mouse model, you get expansion of the CD11B low population. And so this group labeled the CD11B positive cells mammary tissue macrophages and the CD11B low cells TAMs, or tumour-associated macrophages. And it's also been shown that these TAMs are notch dependent, they're capable of proliferation, and they express CX3CR1 and CD11C, and as shown here, they are CD11B low. So this is a really enticing similarity with the DMs that are present in normal development. And of course, breast TAMs are pro-tumorigenic, immunosuppressive, and they aid metastasis. So any connection between these TAMs and normal development will be really important to understand further. We took the data from this paper, which looked at the genes up and down in TAMs. So here we have genes up in the TAMs, and they really strongly associate with the DMs, whereas the genes down in the TAMs are associated with the stromal macrophages, confirming that the DMs are really similar to these TAMs molecularly, as well as their marker expression. Now I looked at flow cytometry throughout tumour development with DMs here, with CD11C, very rare in the normal gland. But then as the tumours develop, you get a really big expansion of the DMs. And then looking by imaging in the CX3CR1 reporter mice, we find cells within the tumour that have a really similar morphology to the DMs in the normal ducts with this dendritic appearance. And looking over a number of mouse tumour models with the normal glands here, and then progression from hyperplasia to tumour. In each of these models, we see expansion of the DMs, but not of the stromal macrophages, showing that this is a really general phenomenon. So to conclude, I've shown you that these ductal macrophages survey the mammary gland across the whole duct system, that they are derived from the bone marrow during puberty, but before puberty and early life, they are embryonic derived, showing that they are essential for epithelial remodeling in involution, and their absence results in the delay of alveolar collapse, and that breast TAMs are really similar to these DMs. So looking at this diagram of mammary gland development, we have the ductal macrophages here within the epithelium, and then the stromal macrophages here, which are outside the epithelium. And in pregnancy, you get proliferation of the DMs to expand and maintain their density on the epithelium in lactation, where they're really closely associated with the myoepithelial cells. And this places them in a perfect location to respond rapidly to the cell death in involution and to clear away these cells. And then their location and morphology is maintained throughout tumourigenesis, where they continue to form a network within the epithelium in multiple cancer models. So finally, I would like to thank especially Jane and Jeff for running a great lab and being fantastic supervisors, and also Anne Rios, who's now running her own lab in the Netherlands, who is a fantastic supervisor and who taught me all of the 3D imaging, which is such a fundamental part of this work and really fun to work with. Thank you to Felicity, Francois, Gordon, and Luke, who were directly involved with this project, and also to Bianca, Jim, Michael, and Ray, who I've worked with indirectly on this project. But of course, also the whole breast cancer lab for being a really great place to work with. And I miss seeing you every day. Thank you to the Center for Dynamic Imaging. It's a really fantastic facility with many amazing microscopes, and this work was so dependent on that facility and all of the bioservices and fax facility staff. Thanks to Bhupinder, who did the RNA work, Scott Mueller, who we developed the intravital imaging with, collaborators at A-Star in Singapore, who we did the MS403 experiments with. Thank you to tissue donors who make this work possible, and to my funding sources. So thank you very much, and I would be really happy to take your questions.
Okay, fantastic talk, Caleb, and some of the best images that seen for a very long time. We do have a few questions. We've got one here. How are the DMs regulating their proliferation during pregnancy? You're able to comment on that?
Sorry, I think I dropped out for a minute.
Okay, the question was, how are DMs regulating proliferation during pregnancy?
That's a good question. We don't know yet. It's something that we'd really like to tackle, but the models that we have for DM depletion are not entirely specific. So we have the CD11C DTR and the blocking antibody, but these also deplete dendritic cells in one case and the other macrophages in the other case. So we really need to find a way to specifically deplete the DMs in order to look at how they might be regulating proliferation of the epithelial cells in pregnancy.
So that my question might be tied to that. You demonstrated towards the end the proliferation of the DMs during tumourigenesis in a couple of models. Have you done or have you planned the experiments of depleting the DMs in those models to see whether it has any impact on the rate or the extent or the type of tumourigenesis that occurs?
Yeah, we spent a while trying to find a way to do that, and we used a model with CD11C Cree crossed with a CX3CR1 Flox DTR to try and really specifically deplete the DMs. But the problem with this is that long-term treatment with DT, the mice develop antibodies against it, so it reduces in efficacy. And the DMs are also able to replenish themselves. So we had success in specifically depleting DMs in short term, but looking long-term over tumor development, we haven't been able to do yet.
From all the knowledge of, the background knowledge, what would you anticipate would happen if the DMs were depleted during that tumourigenesis?
I would suspect that primary tumour growth would be largely unaffected. Perhaps the specific phenotype of the cancer would be altered a bit, but probably more effect when we look at immunotherapy or other treatments, even just chemotherapy, because there's so many DMs within the tumour, they'd be able to phagocytose all of the dying cells. And so perhaps if we deplete them and then use chemotherapy or immunotherapy, then that could be beneficial to deplete them.
Okay, we have a question from Robin Anderson. Great talk. The DM support proliferation of the duct during puberty, but then switch to being phagocytic during involution. What triggers them to switch function?
Okay. Well, there is some phagocytosis in puberty as well, but we think that the DMs are probably not responsible for the majority of that effect in puberty, because the other studies have shown that the other population, the CD206 positive cells, are probably more responsible for that. And in our experience as well, when we try to deplete the DMs in puberty, we don't see much of an effect. So we think that maybe the DMs are most important in involution and perhaps other stages as well. But in puberty, we don't think they have a huge effect, and it might be another population.
But they're just sitting there, not actually functional.
They might have a function, but we haven't been able to find it yet. And it might be something to do with tertiary branching later on, rather than actually progression of the ducts through the fat pad.
Okay. So, a general question about these macrophages and macrophage subtypes. Are this type of resident macrophages found in other organs or other epithelia? Are there correlates in the, in the skin and in the, well, I don't know, the pancreas, I think you mentioned?
Yeah, they are really everywhere. And pretty much every different niche within an organ will have its own macrophages. So there's nerve associated, blood vessel associated. In the skin, there are epithelial macrophages. And it would be really interesting to look in a variety of other epithelial organs, like glandular epithelium, to see if there is a parallel population to the DMs, which I think would be highly likely.
And in the areas where it is, the immunotherapy, you've mentioned a couple of times being the very hot area right now. Is there any suggestion that these DMs play any role in response to immunotherapy?
Yeah, there was an earlier study, the one that determined that Notch was important for their maturation in tumours. So they used the same model that we used, the RBPJ CD11C CRE, to block Notch signaling in those cells. And they saw greater activation of CD8 T cells. I don't believe they did immunotherapy in that case, but there was just a basal elevation in T cell activation.
Okay. All right. Are there any other questions from the audience? You mentioned, and you might have explained it and I missed it, but that one of the roles of these DMs is to, I think you said, promote stem cell activity. I think that was the comment, and I wonder if you could expand on that a bit. Again, thinking cancer, the stem cells and the basal phenotype is one of the remaining challenges. So I wonder what you meant when you made that comment, that promoting stem cell activity.
So that was a study published in 2018, and also Jane and Jeff's group noted that a few years ago when they depleted macrophages, they had less activity of the stem cells. But the more recent publication showed that Wnt signaling from the macrophages promoted stem cell activity. And that was actually downstream of notch signaling. So that really they didn't find a specific macrophage population that was responsible for that, but I think the DMs are really prime suspect for that. We didn't see any Wnt molecules from our RNA-seq, but maybe we need to do a different analysis to find that. And in terms of the basal-like cancer, I can't really comment. I think, yeah, we'd have to get better mouse models for depletion and investigate maybe in human tumours whether these similar cells exist as well.
Sure. Okay. All right. Are there any other questions for Caleb? If not, again, I'd just like to thank you for presenting. That was a spectacular talk, and I, you know, the way that you've taken it from, you know, told the whole story is typical of the work that comes out of the Bisphaetal Lindemann laboratory. But congratulations on it. We have another grand rounds in 2 weeks. It will be publicised. Unfortunately, I can't remember who is presenting, but I'm sure it will be another excellent talk. Thank you everyone for joining, and we'll see you all in 2 weeks. Thank you.